Hello, my name is Koen Beerens from the UGent Center of Synthetic Biology, in short CSB. In this presentation I will give you a short introduction into synthetic biology and how synthetic biology can be used as a sustainable tool for the production of value-added compounds. First of all, let us make clear that biological systems have been used by mankind for millennia, and that the most common examples are of course fermented products such as beer, wine, yogurt and rice wine, but also rennet, the complex set of enzymes produced in the stomachs of ruminant mammals that is used in traditional cheese making. However, nowadays, microbial rennet and recombinant fermentation-based chymozyme, the active enzyme in rennet, are also used in the cheese making process. For example, to make vegetarian cheese. Another example in which recombinant DNA technology led to a great progress in production and applicability is located in the medical sector, namely insulin. Indeed, since human insulin is produced recombinantly in bacteria and yeast, major issues linked with animal insulin extracts, like allergic reactions, skin rashes and low yields, are avoided. Synthetic human insulin has led to diabetes becoming a manageable disease rather than a fatal disease, which it used to be. Recombinant DNA technology is also applied in a much wider context, such as research that is focused on making the switch towards sustainable industry. You might wonder what a sustainable industry means. Well, this means that we want to step away from oil-based chemistry and chemical synthesis routes that utilize, for example, heavy metals as catalysts. Indeed, we want to develop production processes that are both bio-based and cost-efficient, and as such will be able to compete and/or replace the current chemical synthesis routes. Important points of attention are utilizing renewable, plant-based substrates and focus on waste reduction. Therefore, we aim to exploit microbes and their enzymes for biosynthesis of value-added products, which can be summarized under the term synthetic biology. Synthetic biology can be defined as the artificial design or redesign of biological systems for practical applications. This means that we will alter microbes, such as bacteria, yeast and archaea, and their enzymes in our favor and make them perform certain desired reactions. This requires a multidisciplinary approach that combines molecular biology, think about altering DNA, with microbiology and biochemistry, such as expression of enzymes and entire pathways in microbes. In addition, the research fields of bioengineering, bioinformatics and automation are increasingly important as they help us to more rationally design improved microbes and/or enzymes based on the wealth of available natural information, and by speeding up the screening process to detect better variants. Furthermore, our output also becomes input as we apply the design, build, test, learn cycle, in which we learn important lessons from earlier designs, whether they have been productive or may have failed. In the end, we aim to produce value-added products that will find their way in a variety of industries, such as green chemicals, pharmaceuticals, food and feed, agricultural chemicals, as well as cosmetics. Therefore, 3 research groups of the Faculty of Bioscience Engineering have joined forces in the Center of Synthetic Biology. The research group, led by Professor Tom de Smet, focuses on engineering individual enzymes to improve specificity or stability. The research group, led by Professor Marianne de Meij, focuses on metabolic engineering, meaning altering entire microbes and their pathways to produce valuable chemicals. Thirdly, the team led by Professor Inge van Boogaert investigates and engineers membrane transport in microbes, aiming at either improving the uptake of substrates or increasing the export of chemicals that are produced inside the microbe. Their common goal is to develop new bioprocesses for the biosynthesis of value-added compounds with an emphasis on tailor-made carbohydrates, plant metabolites and glycolipids. We focus on integrated bioprocess development, which means that our projects include all the steps from concept development over microbial cell factory or enzyme optimization to finding ideal fermentation conditions and finally downstream processing to obtain pure chemicals. All these steps are done at lab scale, ranging from microliter scale up to around 30 liters as our biggest volume. For further scale-up, we work together with partners specialized in working at larger volumes. At the Center of Synthetic Biology, we combine the knowledge, experience, and expertise of 3 PIs and a team of over 50 junior and senior researchers with state-of-the-art infrastructure, of which the latter is grouped in one of UGent's new core facilities, namely the HTS for SynBio core facility, which stands for High Throughput Molecular Biology and Screening for Synthetic Biology. With the infrastructure and instruments grouped in this core facility, we can perform all the different steps in a typical synthetic biology project, starting from altering the DNA all the way to detecting a diverse palette of products. In other words, all steps in the SynBio chain are covered, making it a complete biofoundry. The most important examples of included instrumentation are our custom-made robotics platform that contains both molecular biology and screening modules, allowing automation of DNA modification, cloning, transformation of DNA into cells, colony picking, and liquid handling, all the way to screening and so-called microtiter plates. And secondly, our coupled analytical systems. These can be applied for the detection of a very broad range of biomolecules, including secondary plant metabolites, glycolipids, and carbohydrates. In addition to these, compatible instruments like multimode microtiter plate readers, fermenters, and bioreactors, as well as all kinds of auxiliary instrumentation that is needed to execute diverse SynBio projects, can and will be provided if needed. In terms of offered services, the Center of Synthetic Biology can help you with all steps in the synthetic biology chain, going from development of synthetic biology tools, method, strains, and enzymes, over process optimization, all the way to the productions. To clarify how we exactly apply synthetic biology, we have selected 2 different examples. The first one will focus on how we apply enzyme engineering for the biosynthesis of rare sugars, and in the second example, my colleague Brecht de Pape will explain to you how we develop biosensors for high-throughput screening in synthetic biology. Finally, I want to end with our contact information in case you want to contact us regarding possible future collaborations or in case you still might have questions for us.